Welcome to the fifth and final session of the PCE3 Community Workshop, where we'll be dealing with peering into the past with today's biochemistry. The topics will be covered by the different speakers, Hannes, George, Moran, Elizabeth, and Greg, spanning genetics, metabolism, chemical evolution, earliest evidence of life, and rewinding life's clock, respectively. Now, taking a big picture into this chemical origins of life, we are concerned with how the inanimate transition to the animate. And we try to draw clues from our own life that exists today. And we have been quite conditioned by this kind of a picture where we see a Darwinian evolution from a simple single cell to multicellular organism, which is easy to understand based on the fossils that we find. However, when you replace these with the constituents of what we consider to be life, the biochemistry and the biochemical molecules that make up life, such as proteins all the way to lipids, we are struggling to reconstruct how it happened in a historical fashion because there are quite large gaps in our knowledge and there are plenty of black boxes in between. And these black boxes reflect our inability to reconstruct a historical picture and not the inability to demonstrate it in a lab. And that is because this is complicated by the absence of intact biomolecules of life from early Earth, where it has been destroyed by various processes and by extinct life itself. You find something and you question whether this is something that would have existed on early Earth. And Elizabeth Bell will be talking about more of these problems in trying to identify earliest evidences of life. And this complicates our effort to reconstruct historically how such processes could have happened on early Earth. Today's biochemistry is made up of proteins, which are the enzymes driving the whole biochemical process, nucleic acids, which store the information and transmit them in between, and translation of proteins. And it's important to note that these are made up of homochiral one-handedness in terms of building blocks. The proteins made up of L-amino acids, the D sugars making up the bulk of the backbones of nucleic acids and various other biochemical pathways. Metabolism is a hugely vast, complex network of reactions, and these are all enclosed in a container made up of lipids, which contains a huge suite of molecules, including the polysaccharides. So if you take the proteins, they are broken down into the peptides made up of these amide bonds, and these are further broken down into the L-amino acids, which are found in extinct biology today. Nucleic acids, which are RNA and DNA, are broken down into their constituent parts, which are the sugars, again emphasizing the chirality of this, and the nucleobases, which are made up of purines and pyrimidines, and the phosphate, which acts as the linker in between. Metabolic pathways, I just show two representative examples. One is the citric acid cycle, and the other is the pentose phosphate pathway cycle, which are made up of carboxylic acids and sugar phosphates, respectively. And the lipids are made up of a general representation of basic phospholipid structure here, which is made up of phosphates, glycerol, and fatty acids. Now, these are the constituents of biochemistry as we know them today. And it's only natural that we use these constituents to try and understand how they would have formed on early Earth and how they would have come together to make the biology and life that we know today. But this biocentric approach also has resulted in focusing only on these building bio blocks. So, for example, life is made up of these biochemical macromolecules. Therefore, we focus on their constituents. And this is therefore a purposeful approach. We know how life is today. We would like to know how it started on early Earth with the respective building blocks. Therefore, we focus only on a narrow pathway of these building blocks, giving rise to these biochemistry, biomolecules, and then leading to life. Now, that kind of a narrow focus and trying to extend biological paradigms into prebiotic chemistry has led to classical theories which claim either one class of molecules appeared first. For example, amino acids are so elementary that they are found in meteorites and in prebiotic reactions in wide variety of conditions. Therefore, the thought process was that the proteins would come from these amino acids, would have formed first, they would act as catalysts, and they would make the rest of what is needed for life, and then life arose. Then there is the current favorite, RNA world. Though the nucleic acids are not as elementary as amino acids in terms of how they are put together, but still, nevertheless, the powerful evidences of running biochemistry backwards has resulted in an RNA world where it argues that RNA comes first and then rest of the molecules are invented later. And the metabolism world argues no, it must have been small molecules that assembled themselves in terms of a reaction network that would have been self-sustaining and then gave rise to the rest of the molecules. And currently, there has also been the lipid world, which has been proposed for all of this to exist in a fashion that they could function together. So, looking at prebiotic chemistry, there are three pillars of prebiotic chemistry that have been argued historically. Even today, people point out to this foremost reaction where a simple molecule like formaldehyde, which could have existed on early Earth, is converted to a suite of sugars. One among them is ribose. The same way, the Miller's famous spark discharge experiment, which gives rise to amino acids from mixtures of reduced gases, and Oro's formation of adenine from hydrogen cyanide, which gives rise to adenine. And these building blocks have a striking resemblance to what life uses today. And therefore, the argument is made they are building blocks of life, and therefore, life must be easy to come together. But what we fail to recognize many times when we make these claims is that many of these reactions, even the ones that are being published today, many of these prebiotic chemistry reactions produce a suite of compounds, not just one among the many. And so to claim that these are building blocks of life, you may be right, but that is not the same as making life from these terrible mixtures that you make. Moreover, the meteorites that are coming from outside are also rich in these building blocks that we saw before. Here are examples showing the distribution of amino acids, comparing it with what we know. And it's amino acids, sugar acids or sugar molecules, nucleobases, fatty acids. And so you are beginning to see the components of what would be, again, building blocks of life. But again, it is to be noted very importantly and emphatically that they are one among many, 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 many compounds. And therefore, what it means to life as such, here or elsewhere, is debatable and questionable. But one has to be careful that we do not take just the generation of building blocks of life either on meteorites or in any reactions that are claiming plausible prebioticity and convert it to this. It is quite distracting when this happens and takes away from the chemistry that we need to pursue in a serious manner. And it is important to note that building blocks of life is not the same as claiming origins of life or life. So let us take an example of RNA and see whether this prebiotic chemistry meets it. Now, these are the building blocks of RNA as we know it today, and the nucleic acids. So if you had ribose from the foremost reaction and the nucleobases from your meteorites or the hydrogen cyanide chemistry, can we put them together? And so far the answer has been, it has been highly inefficient. No doubt there might be in future, there might be some efforts to do this, and it might be something from a clever synthetic organic perspective to do this. But even if it is shown, there is really no connectivity in terms to what biology does. As I will show you later in this talk, just making the building blocks alone is not enough to claim victory towards the origins of life chemistry. Just the prebiotic presence of these in a meteorite or in a prebiotic reaction does not translate or guarantee further reactions. And therefore, the structural similarity you see of these building blocks of life, wherever they may be in a meteorite or in a reaction mixture, does not mean that it is origins of life. However, if you allow for alternative reactants, alternative molecules that would be present, for example, there has been quite a lot of work in RNA building blocks. Right here, as shown by the work of Sutherland, Benner, Carroll, and the more recent Sutherland work, where people have shown that if you start from building blocks that have absolutely no connection with what life does today, you can still make the building blocks of biologically relevant nucleotides. For example, you start here with cyanide, glycolaldehyde, glyceraldehyde, 1,2-cyclic phosphate, amino isoxazoles, and 5-formamidopyrimidines, which life does not use today. But it makes the nucleotides in a plausible prebiotic manner. Therefore, the question is, the type of chemistry that is shown in prebiotic chemistry with building blocks and the type of chemistry that occurs in biology today are based on two different chemistries that don't seem to connect. And therefore, the idea is, did you start out with a different chemistry and switched over to what the chemistry is today? It must be so if you believe in this type of chemistry, which is not used in biochemistry, then there must have been a switch. There must have been the switch. And therein lies the problem for many who do not believe this paradigm because it's not parsimonious. And there is the other view, which says you must be parsimonious going from what life is doing today all the way back. And that is the theory which is driving the metabolism first approach, where people argue that based on such reverse citric acid cycles, which is considered one of the ancient pathways, that it must be based on carbon dioxide chemistry and iron sulfide based chemistry. And people have presented these arguments in a very elegant manner, and a lot of ideas have been proposed. And I would refer you to George Cody's presentation for further details on this. So this approach has also been taken towards membranes, and you will see that there are highly complex membranes, phospholipid membranes today. And people say, since phospholipids may not have been available on early Earth, maybe it's the simple fatty acid-based assemblies that gave rise to that. And there is good evidence presented in the literature from the work of Deamer and many others that fatty acids are capable of forming such cell-like structures. And this is one from the compounds extracted from the Murchison meteorite. As I said, people have argued that phospholipids may not be present, and therefore, again, the question becomes, how does the potentially prebiotic fatty acids transition towards the more complicated phospholipids, and how does this transition take place? Of late, there has been some recent work, and I would refer you to Dave Demas and Christine Keating's presentations to refresh your memory on how complicated this is. Of late, there has been some work that suggests that phospholipids may be involved from the beginning. So, these views are subject to change as more and more facts come to light. Overall, starting with biochemistry, it's a biocentric approach and leads to, these kind of model systems where one, or the other biochemical molecule is proposed to come first. And then the rest is invented. It has a strong biochemical bias, which is based on parsimony, and it results in experiments that are designed, model experiments that are designed to maximize the production of biomolecules under prebiotic conditions. And by definition, it ignores the other prebiotic molecules that are present. For example, remember the meteorite, where you just focus on the ribose or adenine as building blocks of life, but forget the rest of the inventory. And therefore, many have argued that these approaches have little or prebiotic relevance and they are model systems because they do not take into account the full complex history of early Earth. And it should be pointed out, none of these models have gained universal acceptance among the larger community. People are still divided into one camp or the other, arguing strongly for one over the other. The current favorites right now are the RNA world first and the metabolism first theory. And right now, there is a realization that maybe rather than having one invent the other, why not mix them together? And this is the systems chemistry. And this systems chemistry is a term invented by Kedrovsky, and it is mixing these systems once they are made separately. And I must emphasize that there are plenty of reviews which talk about this, so I will not go into detail, but it's mostly at the level of the monomers, mixing the monomer together with another monomer class of compounds and see what happens. And there are also plenty of experimental approaches that I show here, and most of them deal with, again, the mixing of the monomers. Of late, people have been also worrying about what happens at the oligomeric level. And here is one example of what happens when you mix RNA and DNA in an oligomeric backbone and how useful it might be towards the emergence of RNA and DNA. There are practical considerations, and this systems chemistry approach is severely limited by the highly complex nature of the results that would result, from mixing too many compounds and allowing too many parameters to dictate what sort of chemistry. A classic example is the Formose reaction, which produces a whole suite of compounds that are very difficult to analyze and difficult to control. So if you take this biochemistry approach very seriously, we have to ask the difficult and important question: how do you use this to guide reactions as to what reactions would take place when there was no such biochemistry present? Can this over-reliance on biochemistry also be misleading us? And I will give you a concrete example here to argue that it might. For example, in RNA, there are two pyrimidines, the cytidine and the uridine, which people argue, because the same building block of cytosine and uracil is formed from simple molecules like ammonium cyanide, there must be some sort of a connection. This is the usual argument that is made. But if you look actually into how biochemistry makes it, biochemistry does not synthesize uracil or cytosine. It makes orotidine, which comes from orotic acid, which comes from aspartate and amino acid. As I pointed out before, everything is amino acid based on sugar chemistry in biology. However, it is also of interest to note that the same orotic acid is also produced in prebiotic chemistry. But people do not focus on orotic acid because it's not present in the final product RNA. Therefore, we look at the final product RNA in biochemistry, and then we look at what constituents are given from that, from prebiotic chemistry, and try to make a straight line. And we know that might be misleading because biochemistry does not synthesize any of the four canonical nucleobases till today. The only nucleobase that is synthesized de novo fully is the orotic acid, and it's also present in prebiotic chemistry, but we seem to ignore it completely. And the question is, why is orotic acid not present in RNA? And it must be something to do with its base pairing property. And work has been done to show that indeed orotic acid is a poor base pairing partner. This curve represents a nice Watson-Crick duplex, which is destroyed when uracil is replaced by orotic acid, even a single substitution. And the reason is that there is quite a lot of hindrance with this 6-carboxy group, which rotates the pyrimidine away in such a fashion that the Watson-Crick is facing the wrong way. Therefore, there is no base pairing, and it is detrimental in RNA. Therefore, the argument is, does this not mean nature selected a better molecule? Is that not part of evolution? The answer would be yes, but the problem is, if you go and use that argument, you will have to make that argument only at the level of an oligomer or a polymer where the base pairing can take place and in the presence of a complementary base pairing partner. Without those two, you cannot make that argument. And the question is, how would you do this when you just find orotic acid or uracil in a meteorite or prebiotic chemistry and claim that is the origins of life? Because this is all true for each and every part of RNA. And that means the selection of RNA did not take place just because uracil was formed in a meteorite or it was formed in a prebiotic reaction, but you had to wait till the oligomer or the polymer of RNA completely emerged. And that is shown in this slide. So if you have a mixture, and this is the mixture you are seeing in an exoplanet, how would you, without any knowledge of what that chemistry is, select, ribose is the one that I would want, or that is the one that is going to end up in the final product? How do you select, or how does one make the determination that this is going to be the part? Do you select here, or do you make the selection at this level? You have to wait until a functional biomolecule comes out, which is useful for a certain function. And you cannot make that determination where there is no such selection criteria because that function has not yet appeared. Therefore, you have to be careful what you can select on in these mixtures. You can select on different other properties such as solubility or stability, or what happens to those molecules when they crystallize out, or things like that. But you cannot select for a function that has not yet appeared, because if you try to do that, then you're imposing a purposeful selection, and that cannot be done, because as somebody elegantly said, evolution is a tinkerer, not an engineer. And that is also true for chemical evolution. Therefore, peering into the past with today's biochemistry can also be misleading if we focus too much only on what is in biology today, because the prebiotic chemistry and prebiotic inventory is quite vast, and they have to be included, and you cannot simply ignore the prebiotic platter. But if you do take that into account, the non-biomolecules, can they play a role? Could they have played a role? Can the bottom-up approach complement the top-down approach of using only biochemistry? So the question then we can ask is, can we peer into the future with prebiotic chemistry to compensate for the shortcomings that are associated with using only biochemistry to peer into the past? We'll take an example. The emergence of peptides has always been dealt with using only amino acids, because amino acids are found in meteorites and in prebiotic reactions. However, it has been quite a challenge to take pure amino acids and take them over to oligopeptides, in spite of the publications that are appearing today. For the simple reason, there are no scenarios on early Earth that produce only the amino acids or derivatives thereof. It is always present as a mixture. So if you can take the other compounds that are present and see whether they would help in the formation of peptides, then it becomes a little more meaningful to talk about origins of life from a mixture of compounds. This has been shown, in fact. People have argued that hydroxy acids are all present in these prebiotic mixture reactions. For example, the very famous Urey-Miller experiment produces a lot of hydroxy acid along with amino acids, and they are also present in meteorites. So the question is, why are we ignoring the alpha hydroxy acids and focusing only on the amino acids? Again, that is because we are focusing on extant life and the biochemistry and peering into the past using the today's biochemistry. But more importantly, it will not get you an impactful publication, or it will make headline news, because imagine if Stanley Miller had said, I produce alpha hydroxy acids in my spark discharge experiment, we would be not talking about him as the founder of prebiotic chemistry. The work that has demonstrated that alpha hydroxy acid presence actually leads to peptides is quite interesting. So this is work that was done showing that if you start, instead of only amino acids, a mixture of alpha hydroxy acids and amino acids, the natural chemistry that takes place is the formation of an ester bond from this hydroxy acid, which then enables the attack of the amino acid on this ester bond to give you a peptide bond. And this leaves a free carboxylic acid always at the end, which then can continue this same esterification followed by attack of the amino acid. And this continues, and you end up naturally, because of the thermodynamic and the kinetics of the reaction, into a peptide enriched polymer. And this has been shown by doing the mass spec analysis of this reaction mixture, where the glycine alone does not polymerize, but the lactic acid alone does and makes a lot of polyesters. And if you mix them together, you begin to make deoxypeptides. And these deoxypeptides can be analyzed. And by mass spectroscopy, and the fragmentation pattern clearly shows that you have enriched the middle of the polymer with a lot of peptide bonds. For this, I would refer you to Luke Lehman's talk, and also to Moran Frankel printer's talk that is going to come later this week. So the idea is that instead of focusing on unrealistically only on amino acids, which is not plausibly prebiotic in terms of having only pure amino acids, and which has led to a lot of work, but under prebiotic conditions, being very difficult. Rather, if you allow for the mixture, which is naturally present in meteorites or in the potential prebiotic reactions, you actually make it much easier to go to the same peptide bond that is used in biology. And the nice thing is, there seems to be, intriguingly, quite a good correlation with what biology does today. In the ribosome, which makes proteins, biology does exactly the same thing. It makes an ester bond, and then the amino group attacks from the tRNA sitting next to it and makes an amide bond. It is the same ester-amide exchange that you see in prebiotic chemistry. It's also true for the non-ribosomal peptide synthesis. Therefore, by considering what other molecules that would have been prebiotically plausible in that reaction mixture, and not focusing only on amino acids, it leads to one possible solution that naturally leads to peptide formation without actually wanting the peptides to form. However, I must point out this is only a proof of principle experiment, because this has not taken into account the other molecules and other prebiotic clutter that would be present. And this is again pointing out to the limitations of the systems chemistry approach of how quickly things can become complicated. Therefore, rather than insisting on prebiotic chemistry be the same all throughout to biology, you can allow for mixtures of prebiotic acids and begin to see whether there would be a different type of molecule that emerges, which then switches over to the other one that biology is using. But there is some similarity in terms of the mechanisms and the type of molecules that biology uses. So this kind of approach may be quite useful, complementing the top-down approach with top-down approach with the bottom-up approach, and seeing where we can learn from each other. The peptides, are emerging from this kind of chemistry that I showed. But what about the RNA world? The RNA world insists that protein are a later invention after RNA came about. And they have very good reasons to argue that based on extant prebiotic chemistry. One problem with the RNA world hypothesis is that the definitions keep changing, and it has morphed from an RNA only world to now an environment where RNA is acting both as the catalyst and the genetic material, and then RNA hands over the tasks to proteins and DNA respectively. And this is based on the smoking gun that many people point to is the ribosome, where the action center is the formation of the peptide bond, where no other peptides are present, no proteins are present, only RNA and other things such as cofactors that are present, which have RNA nucleotides structures in them. However, there is a completely opposite view, which says the same ribosome shows RNA and proteins co-evolved, and RNA did not invent the protein synthesis. And there should be other components in there, not only just the ribose-based RNA, but other sugars in the backbone or other type of linkers in the backbone. And Moran would be talking about this in her chemical evolution, where the idea is to bring all sorts of different, possibly prebiotic structures into the mix to see what naturally emerges. So about the nucleic acids, we in the peptide we showed earlier that mixing of amino acids and hydroxy acids naturally leads to the formation of peptides. Can a similar approach be taken towards nucleic acid? Can a mixture of these prebiotic building blocks give rise to a homogeneous RNA backbone that naturally arises out of interactions that would develop throughout a systems chemistry approach? There are people who have argued that RNA is too complex to come about by itself, and there must have been simpler pre-RNA alternatives which gave rise to RNA. And there has been now some work which shows that these kind of mixtures can give rise to RNA, provided you allow for a replication machinery to help in the selection in terms of what templates and what monomers can be selected. There has also been recently experimental evidence giving credence to a very old idea that RNA and DNA might have actually appeared together, and there was no necessary for RNA world to invent a DNA world later. There is actually quite a good argument that a mixture of RNA and DNA helps the replication much better than a pure RNA itself. And I would refer you to these two papers where it has been shown that mixing and not having a pure RNA backbone actually helps RNA replicate much better than a pure RNA system. Hannes Mutschler would be dealing with this section in more detail. Coming to the end of the presentation, what we see from the two different approaches, peering into the past with today's biochemistry and peering into the future with prebiotic chemistry, there seems to be this Goldilocks story. The biochemistry inspired models seem to be too clean and too structured for many other people to accept it as a universal model of origins of life. Whereas the prebiotic chemistry inspired models seem to run into the difficulty of being too messy and intractable to analysis. So the question is there a habitable zone equivalent of prebiotic chemistry that the early Earth environment can provide, where you don't run into this messiness, but you are not that as clean as buying chemicals from Alrich and putting it in a round bottom flask, but you are able to come up with a model system that would be able to straddle both extremes. This leads us to the question of how prebiotic chemistry gives rise to biology and life. And as we saw, there might be a value in not trying to run the same chemistry throughout, but allow for chemistries to change and evolve as environments change. And this is pretty important because the biology and life we see today is contingent on the environmental conditions such as pH, water, etc. You change any one of those, you might actually change the trajectory and not end up in life as we know it today, but an alternative biochemistry and alternative life. And that tells us the difficulty in using biochemistry to peer into the past, because the molecules that we have in biology today are not a destiny, but a destination. And keeping that in mind, I summarized my talk here saying that peering into the past with today's biochemistry was a highly logical place to start, but it has led to model systems even today, because using that as the sole guide has ignored the prebiotic inventory and focused only on the biomolecules. It needs to be complemented by peering into the future with prebiotic chemistry, such that we take other molecules that could have been present along with biologically relevant molecules and see how they would help in the emergence of the type of biopolymers that we see today. However, it needs to be emphasized, the bottom-up approach can be severely limiting in terms of experimental complexity, and therefore one has to know how to balance the cleanliness of the model of the top-down with the messiness of the model from bottoms up. Leaving this aside as an end, the question is, all this biochemistry that we talk about must have led to some sort of early life form. What is it? And how can we understand that, in terms of, again, using the today's life, looking backwards? For that, Elizabeth Bell and Greg Fournier will be making presentations dealing with those kind of things. And I leave you with this slide as a philosophical way out of this conundrum, that there is not too much exuberance when you see a paper appearing in today's journals to say that origins of life has been solved. At the same time, not to be too skeptical that it can never be solved, because as Albert Schmoser said, and this is probably the central message for my talk, would be that the origin of life cannot be discovered, it must be reinvented. Thank you.